I am thrilled and so appreciative, David, to welcome you to this forum. David is one of the world's most famous scientists and entrepreneurs, best known for understanding why we age and how to reverse it. He's a New York Times bestselling author, a tenured professor of genetics, and co-director of the Paul F. Glenn Center for the Biology of Aging Research at Harvard. He's best known for his work on genes and small molecules that delay aging, including the sirtuin genes, resveratrol, and NAD precursors. If you have questions, which I'm sure you'll have many for David, please send them through the Zoom question and answer link at the bottom of the page. From 10:30 to 10:45, we'll host a Q&A session and answer as many questions as we have. But most importantly, David, I can't tell you how appreciative I am that you are joining us today. We had just the best time in Palm Beach, and we so welcome you into our community. Thank you for being with us.
Well, thank you, Carolee, and thank you everybody who's joining this morning. Yeah, it's great to be here. This is not really my library. This is a fake library. I'm actually in my basement, which is the only quiet place in my home. Yeah, so today I have just a short amount of time to tell you about some new work that we've been doing. For those of you who really don't know anything about my lab's work or about the field of aging research, I'm here to tell you that the field has come a long way in the last 20 years. We started out studying little organisms like yeast cells and worms. Through the 2000s, a lot of us, myself included, we worked on mice, and now we're at a place where, in a time where there are dozens of human clinical trials using the discoveries out of the aging field to treat diseases of aging. Because while we give names to various diseases, Alzheimer's, various dementias, cancer, heart disease, frailty, etc., really we're just naming the effects of aging, right? To give you an example, the one I like is cancer. If you smoke, you increase your chances of lung cancer by 5-fold. By aging, you increase it more than 100-fold. So, the aging really is something that we've neglected for many, many centuries, actually. And unfortunately, in our society, because we could never do anything about it, because we really didn't understand it the way we do now, we've tended to look at aging, the aging brain, the aging organs, as really just something that happens naturally. And when it happens naturally, unfortunately, we tend to think of it as acceptable. But I'm here to tell you, A, we can do something about it, and B, it's not acceptable to be sick in old age. Let me just turn on my light a little bit brighter. You might be able to see me. There you go. Now you can really see my age. All right, so Carolee, is it okay if I start some slides and share those?
Yes, of course.
All right, some disclosures down there. There's a full list of who I work with at that website. Yeah, unfortunately, the aging research field gets a lot of negative attention, and that's mainly because there's a lot of people who claim they know about aging, and they're selling things that they claim to slow aging, and almost all of it is not based on real science. We've come a long way since the days of antioxidants. We actually now have a whole set of genes that we understand in humans control how long we live and how susceptible we are to those diseases that I mentioned as a result. And so the science that I'm going to tell you about, and those of my colleagues, which I won't have time to mention, unfortunately, though there are hundreds of them around the world, their work is being published in the world's leading journals. There are journals like Cell, Science, Nature, and there's at least a dozen more that I could mention. These are journals that are considered real breakthroughs that change the way we think about biology. Up there with discovering a new planet and new physics. The science of aging is up there with those and is considered one of the hottest areas of science today. And I want to share with you some of the breakthroughs that have been made in my lab just recently. And you're actually hearing about this just maybe a few weeks ahead of some of the work that's going to be published in one of these top 3 journals. So I'll give you a sneak preview of that. And so traditionally, and I would say most people that you would talk to, including doctors, think of aging as something distinct from age-related diseases. And as I've started to tell you, that's actually just factually wrong. The only difference the only real difference between these two is that we give them different names. In reality, what's going on is that aging drives age-related diseases. We don't get dementia typically when we're a teenager, right? There's a good reason for that. And finally, we're starting to understand why we get these diseases late in life and not early in life, right? We take it for granted that this happens, but has anyone really asked why is it that these diseases occur late in life and not so often early in life? And I think the pandemic that we're experiencing really highlights the fact that there are a lot of differences between young and old, men and women, and that if we were able to either delay or in some cases reverse the clock of aging, then we literally wouldn't have a pandemic. And so aging is a really important topic. And there's a real revolution in the field going on that you may not have heard about. And that's why I appreciate the chance to give seminars such as this one, because if it's filtered through the media, and newspapers are often the biggest culprit here, they take our research and they just hype it or they misinterpret it. And the public who doesn't have access to scientists, or at least until recently didn't, really has no idea what the truth is and what's good science, what's bad science. So I'm happy to update you directly. So what I want to cover really in the next half an hour is answering these questions. What is aging? Not just what does it look like? We know what it looks like, but what is it at the molecular level? What drives it? Can we measure it accurately? And can we even slow it? And then really what I want to focus on is the new science of aging, which is showing that at least in the lab and in model animals such as mice, it's actually surprisingly easy to reverse aging. If I had said that phrase 10 years ago, I would have been probably kicked out of Harvard or at least laughed out of a room. But I can now say this with not just a straight face, but proudly, that we are in my lab and a few others around the world showing that you can reverse aging, period. Now, this is one of the more complicated slides I'll show, but it's very important. We as life forms have enormous plasticity. So what do I mean by that? Well, it was discovered by John Gordon, going back in the 1970s, that if you take the nucleus, the DNA, out of an adult and put it into an egg, in this case, he was using tadpoles and frog eggs. But it's true, we know we can now clone pretty much any animal we want. What he showed is that you can reverse aging. You can reset the clock and start life fresh. Now, this isn't really what we want to do for ourselves. It's not going to cure Alzheimer's disease easily. We can't just take the brain out and restart a fresh brain and put it back into you. That's science fiction. But we're doing something similar to that now in my lab, and that's what I want to tell you about. And our research is based on this second line here, cellular reprogramming. And Shinya Yamanaka, a Japanese scientist, going back now over a decade ago, showed that if you add a certain number of genes into adult cells, they might be a skin cell from you, or really any cell you can take from an adult, you can reprogram those to be stem cells. We call these iPSCs, or induced pluripotent stem cells. And it's a real revolution, because now we can do things like take a skin sample or just a spit sample, grow those cells and make mini organs or stem cells that we can put back in the brain, and hopefully regenerate parts of the aging brain, as well as skin and other tissues. And Shinya Yamanaka and John Gordon both together won the Nobel Prize for those discoveries in 2012. Other things worth mentioning are that other life forms can regrow parts of their body very easily and are immortal. Hydra, can do that. It's a very tiny little waterborne organism. Axolotls or salamanders, you cut their limbs off, their hands off, they can regrow those. And we can even regrow large amounts of our liver. The majority of our liver will grow back if we take it out, which is great for people who lose part of their liver. But what it's also great is it tells us that the ability to regenerate and be young again is still in our body. We just need to tap into that mechanism. And I think we may finally have figured out how to do that. But first, let me talk about one leading hypothesis as to why we age. Now, in deference to my colleagues, there are a lot of ideas about why we grow old, but we do believe that the same mechanisms that cause the liver to age and our skin to age are the same underlying processes that cause the brain to age. So if we can understand the causes of aging, it will have a universal benefit to all diseases of aging. That's really what drives us. And one of the leading hypotheses is the following. So this is a representation on the left of a young nucleus, and the black lines are DNA. You can see them under the microscope as chromosomes. And there are proteins that bind to chromosomes and switch genes off. And then here there's a gene that's being read by the cell. That gene is switched on. And that's great. That keeps a young cell functional. It can divide, it can grow. If it's a nerve cell, if it's very young, it can even regenerate. And in fish and in mice, well, typically fish, you can damage the eye, for example, and it will grow back parts of the eye. And we don't have that function anymore as we get older, but a very young baby still has some of that capacity. And the idea is to find out why that happens. And what we think is going on as we get older is that nerve cells and other cells in the body, they lose this nice pattern of what we call gene expression, such that genes that should be off in this particular cell, let's say it's a nerve cell, will start to come on. And conversely, genes that should be on to make that nerve cell a nerve cell will get switched off. And so what you get is a loss of identity. The cell doesn't realize anymore that it's a nerve cell. And no wonder the brain doesn't function efficiently if the genes that it needs to function are not turned on and off correctly anymore. And one of the questions we've had is, does this occur? The answer is yes. But is it an effect of aging or is it a cause of aging? And we can actually test that in the lab. And what we wanted to do was to drive this process. And if we're right about this, we should get acceleration of aging. Now, we don't want to do that to a person, of course. That's really the last thing that we want to do, of course. But we can do this in the lab. And I want to give you one of those experiments, a demonstration. One of the analogies I like to use is a compact disc. Now, when I teach at Harvard, I have to explain what a compact disc is, but I think most of us will know that this is a digital format of music and movies, but they used to get scratched pretty badly, and then the reader would skip, similar to a broken record. And what we're talking about here is the biological equivalent of polishing that CD, that compact disc, so that we could recover the youthful music of our lives and get the body to function like it's young again. And really what it boils down to is my hypothesis, and some of my colleagues agree, is that aging isn't a loss of genetic information. The music is still there, but the scratches occur so that the body cannot read that information correctly, which is good news because you can polish the CD, but if you break a piece of the CD off, you're not going to get the information back. For modern times, it's similar to rebooting the hard drive or the memory, the RAM, and get the software to upload again. That's really what we're trying to do for the body. And we now think that that's actually possible, that there is a memory of an unpolished CD in our cells, and we can press reboot. So just a little bit of biology with some, this is a metaphor for how our cells package our DNA. What we do in every cell so that we can fit 6 feet of DNA into every cell is that it's wrapped up tightly in these spools. Now, these are, of course, not plastic black things. These are actually histone proteins that package up the DNA. And when you have very tight packaging, the gene that will be switched off and remain off. But if you open up in a nice, beautiful loop, let's say here as shown, maybe here, this little bit of loop here, this could be a gene that is switched on. Now the cell has access to that DNA molecule and can, what we call, express it. What we think is going on during aging is that this becomes untangled and messed up, and so that the cell doesn't read the DNA the right way. So the way the cell actually does this is more realistic representation of how DNA looks in the cell. If you could go in there, you've got these histone packing proteins, and the DNA wraps around it about 2.5 times. And this, if you fold it up layer upon layer, which you can see happening over on the top left, eventually will give you a chromosome, which you can see under the microscope. You can't see DNA molecules under the microscope, but we know it's there, of course. And one of the ways the cell packages up the DNA tightly to turn a gene off is to add a chemical called a methyl. And the process is called methylation. And methyls are everywhere. They're in most drugs. It's just a hydrogen atom with 3 carbons attached. But the cell uses this chemical modification to tell a cell that it should be a particular type of cell. Let's talk about the neuron. The neuron, when you're young, becomes a neuron, and it has to stay that neuron in your brain for the next up to 120 years. And one of the ways that it does that is this chemical methyl gets attached to the DNA and stays there. But one of the things we're finding is that this pattern of methyls on different parts of the genome, on different genes, changes over time. And we actually think this is one of the underlying reasons we get old. One of the reasons we believe this is our work thanks to really a couple of leading labs, Stephen Horvath and Greg Hanna, who showed about 7 to 10 years ago that you can read these methyl groups with machines and derive with artificial intelligence an age of the body. And it depends on the pattern that you have at the time. And we can do this for animals, and we can do it for people. If I took your blood, I could quite easily do what's called a DNA methylation clock analysis. And I could tell you that where you are for your birthday candles might be misleading. You might be, you might be average, but you might be older for your actual chronological age, or your biological age may be younger than your chronological age based on this clock. And we believe that this is an indication of true biological age, because we can also use this number to predict when you're going to die. And it's surprisingly accurate. Now, if you don't exercise, you don't do the good things. So you don't exercise, if you don't eat well, you're always full, you've put on too much weight, or you smoke, you actually will, on average, if you look at a population, see an increased rate of aging of this clock, and vice versa. So this doesn't prove that it causes aging, but it's a really good indicator of the rate of aging. And there are actually a few companies popping up that can measure this for you. But getting back to the cause and effect, we wanted to cause the scratches on this compact disc and see if you get aging. Now, this is a mouse that we didn't scratch. It's 16 months old, which is a late middle-aged mouse in my lab. And we had a way to scratch the CD. What we did was we broke the chromosome at different places when the mouse was young, 5 months old, let them heal, and then asked, did that change the structure of those loops and bundles that I talked about? And the answer was yes. We could change what we call the epigenome, which is those bundles and loops. And we got a mouse that looked like this. Now remember, these mice have the same genome, same DNA, but one has accelerated aging. And this mouse and hundreds of them, they're the same age as their siblings, but they undergo this rapid aging. And we've looked at the molecular level, histological level, and even the DNA methylation clock. And these mice are definitely older by about 50%. So can you reset the clock is the question. What if you could hit a button and make the brain young, or make that mouse look young again? It would be the equivalent of taking the DNA that's all untangled and putting it back to this structure. A pretty difficult job, right? And we've been working on this for many, many years to try and figure out how to do it. But we stood on the shoulders of Shinya Yamanaka, the Nobel Prize winner. And I also here want to give public credit to Juan Carlos Belmonte, who's at the Salk Institute, who did a study that's remarkable. He turned on the Yamanaka genes, those 4 genes that I talked about, that would normally make a stem cell. He turned them on in a sick mouse and made it live 40% longer. And it seemed to be rejuvenation when he did that. Now, the problem was that the mice would, if he turned them on for more than 2 days a week, they died. So this is not going to be a medicine anytime soon. But it was an interesting proof of principle that you might be able to perhaps improve health and maybe turn back the clock. Now, what I'm going to tell you about are some experiments in nerve cells. We chose to work on the nervous system because while adult neurons don't grow very well, break your spine, damage your eye, or your brain, it's not going to grow back well. But young brains do, young eyes do. And we thought if we could reprogram, in this case, we're going to talk about the back of the eye, part of the central nervous system. If we could reprogram that to be young, we might be able to regrow the optic nerve and even regain vision in old mice. So we did that experiment. This is Shinya Yamanaka at the night he won the Nobel Prize with royalty in Sweden. So let's get back to this point here, is that Shinya showed that Dr. Yamanaka showed that you could put these genes into an adult cell, these 4 genes, which I abbreviate O, S, K, and M, and get these stem cells. Now, what are these genes? Well, they make proteins that tell other genes what to do. They tell other genes either to switch on or switch off. So we call these transcription factors. They're basically central coordinating gene control, protein producing genes. All right, so these can reset the age of a cell back to 0. But we don't want to reset our bodies back to 0, or we'd have a giant tumor, or we'd die. So we wanted to find a way to do this safely. And a graduate student in the lab, Wancheng Lu, who's now graduated, found that this particular combination of 3 genes, leaving out this MYC gene, was the winning combination. It's seemingly very safe, and it doesn't cause tumors. This was the bad actor here. So we put these genes into animals, and we're going to be putting them hopefully into patients within the next couple of years using what's called an adeno-associated virus. Now, it's not related to coronavirus, but it does look similar, as you can tell. And it's a very safe and FDA-approved way of doing gene therapy. And these viruses are used currently to treat sickle cell anemia and reverse genetic diseases, such as diseases that cause vision loss. And it's working really well. And so we took this virus, we put our 3-gene reprogramming combo into it, and now we could infect the eye and see what would happen. And so what we did with the mice was we damaged the optic nerve. This is in another lab, actually, collaborated with Zhigang He's lab at Children's Hospital. And Zhigang's goal is to help with spinal injury. And this is a way of testing that. So if you don't treat the optic nerve, what happens is the nerves will die back. And you can see here that the dye in orange doesn't flow down these dead nerves. But what we did with our virus, we could now turn on these, what we hoped were anti-aging factors. And if we were right, we should get regrowth. And so we hit the jackpot with this experiment. Turning on the reprogramming, turns out, did reverse the age of these nerve cells. It didn't cause any safety issues. We don't get tumors. But we do get the regrowth of these nerves from the eye back to the brain. And the longer we leave it, the better they grow and the further they grow. And so you can also see that the nerves that were close to the eye were preserved much better than those that weren't treated. So this was a first Hail Mary experiment. We went on to test glaucoma. Again, we're putting in viruses after we've created what's called intraocular pressure, or pressure in the eye. So this is our mouse model of glaucoma done with Bruce Cassandra's lab in Boston. And now we can test, can you not just restore the electrical signals in the eye, but can you actually get physical, functional vision back in a mouse that has become blind? And we did this for 4 weeks, and we had a look at the function. First of all, we did get the electrical signals back. This is before turning on the reprogramming and after. We have shown that this is due to an age reversal process. And we can test vision. What we do is we put a mouse in front of moving lines in an LCD screen. And if the mouse moves its head, we can tell that it can actually see those lines. And we can test how well it can see by changing these lines' thickness. And what you can see in this early experiment that we did with Bruce was that we got vision back. This is without treatment, and this is with treatment. So some of these mice actually got all of their vision back, and some got an intermediate amount of their vision back. But this is the first time that we've actually been, or anyone's been able to reverse vision loss in this model of glaucoma. So I wanted to know, what about regular old mice? Can we reverse aging in the old eye? And this is also, I hope you can appreciate, a surrogate for what we could do for the brain as well, because we're talking about rejuvenating neurons. Now, this was an old mouse that couldn't see. It's not moving its head. It's essentially blind. But we could take a different mouse and we could turn on the reprogramming factors. You might ask, well, David, how do you turn on this reprogramming? Well, we just give an antibiotic. We give doxycycline, which you can take for, resistance to malaria. You can take it for an upset gut. It's pretty benign. And we've engineered the virus so that the antibiotic turns it on. So we do this now for 3 to 4 weeks in old mice. And I think you can probably guess what we saw. We had blind mice seeing again. Now, excuse the little droplets, that happens naturally as the mouse sit there. But the important point is that this mouse is now moving its head. The nerves at the back of the eye are now young again, and they're functional. So those neurons in the eye, I can tell you, we've proven that those nerves remember again that they should be nerves. And in the old animal, they had forgotten how to be nerves. So this is pretty exciting, right? That we can take quite a complex tissue, arguably one of the most complex tissues in the body, the eye, turn back the clock by about 70 to 80% from what it was. These mice, if you're wondering, are 1 year of age. We take it back to a few months of age. And it works again, just like they were young. And what we believe, my colleagues and I, is that we can reprogram other parts of the body, the brain, the spleen, the kidney, the liver. So the field is just beginning, and some of the unpublished work that I was hearing about at a recent conference said that it's possible to reverse possibly all parts of the body. Now, this is a mouse. We don't know if it's going to translate into people, but we're really optimistic because these factors that we're using are already in the clinic for other purposes, such as using them to make stem cells. And we know that this system works in human cells. We've done that in my lab. We can take human neurons, damage them, and fix them with this system and make them regrow. So this is, a bit of tongue-in-cheek. This was a cartoon that was made after Belmonte's paper came out a few years ago. And the idea is that we could perhaps, for the first time, have a handle on not just why we age, but doing something about it. So this is a group of people that I took from a nursing home and reprogrammed them. I jest, this is actually my lab members. They're just fantastic, and it's been a joy to work with them. And it really just, I have one of the best jobs in the world mentoring these folks who, many of them will go on to be leaders in their own right. I do want to point out a couple of people. Where's Wan Cheng? Where is he? Don't tell me he's not in this photo. He might not be. So Wancheng Lu, fantastic guy. He's not here in the photo, unfortunately. Oh no, there he is. Excuse me. He was right up the back. There he is. Also, Daniel did bioinformatic work. And then Xiao Qian, who I can honestly say isn't in this photo, did a lot of work as well. We've got Maeve, who helped a lot as well. And Carolina is also a co-author on this paper. Oh, Alice. Yeah, excuse me, I threw this photo in at the last minute, but Alice is our mouse wrangler. So a lot of the mouse experiments looking at the ability to reverse aging of the immune system and mice running better with some of our other experiments. Alice has been great for that. And thanks to everybody in the lab. So what can you do? Just in the last few moments, I know if we have time for questions, there's going to be a lot of them about this. If there's one thing that you can do of all the things that I think are possible, it would be to eat less often. Now, of course, this isn't about malnutrition. It's not about, certainly not about teenagers eating less. That's not the point, and I would hate to give that impression. It's about people who are my age, I'm 51, whose metabolism has slowed down, and we're still inclined to eat 3 meals a day with snacks, and I think that that's a mistake. And we've known for millennia, actually, that skipping meals is actually beneficial. So I skip breakfast. Some people prefer to skip late dinners, but I think it's scientifically proven now that we do overeat in our society, and those civilizations, and well, cities at least, that abstain once in a while actually do have healthier lives and live longer. So that's one tip. There's quite a few. I wrote a book because I often don't have time to talk about what to do. It's this book. If you want to pick it up, you're welcome to. It's out since about a year ago. And if you don't want to read the book, you can actually just skip to page 304, which is the cheat sheet for what I think is worth considering to have a long, healthy lifespan. It's, you know, I do what I say. A lot of these things I'm doing myself. You know, in part because I think it would be hypocritical to advise to do things and not do them myself, but also because I'm a scientist and I'm monitoring myself as time goes on. I've been doing it for about 15 years, and so far so good. If I see anything dangerous, I'll let everybody know, but so far I'm doing well health-wise. And so is my father, by the way, who's 81. So Carolee, I'll leave it there, and I'll turn it over to you.
Well, David, thank you. I mean, I so enjoy always and so fascinated clearly by your work. A couple of things. First of all, Lifespan is an amazing book, and I wouldn't skip to 304. Every page I thought was fantastic and insightful in terms of information that I think all of us are somewhat puzzled by, but certainly interested in. So some of your early work was in resveratrol. And I know also a couple of people have asked me about metformin and I think it's NMN. If you could talk about some of the different lotions and potions, if you will, that I know a lot of people are taking. I mean, for many years, a lot of us were fixated on HGH and some other things. So, and I'm not suggesting that I'm not recommending or suggesting that either I or others should be taking all of this, but clearly, there's been a lot of research that has shown that these are some things that might retard or slow aging or in fact reverse it. Could you talk a little bit about that?
I can. First, a disclaimer, I'm a Harvard professor and I get criticized when I talk about what people should do. So I'm definitely not saying what people should do. But I do feel an obligation to educate. I know more than most about these medicines. And so I'm happy to give my scientific opinion. So let's start with metformin. Metformin has been on the market for decades. It's been in hundreds of millions of people. It's typically prescribed as a frontline medicine for type 2 diabetes or high blood sugar. And it works remarkably well. Now, it's not perfectly safe. It can have some side effects, but that's also very rare. It's a drug, so in the US and most Western countries, you need prescription. But what's been found about metformin science-wise is that if you look at over 10,000 people, their health over a number of years of taking metformin, there is apparently a protection not just against deleterious sugar levels, but also less cancer, heart disease, Alzheimer's, and frailty in those people who are susceptible. And to me, that's really compelling. There's been a number of studies now that show this. Now, that's not proof, that's an association, of course. But given that metformin is cheap, it's on the list of the World Health Organization's essential medicines for humanity, and the risk is relatively low, especially if you do this under doctor's supervision and you monitor things. I've on occasion been taking metformin myself to see what would happen. But more importantly, I think that we need to do actual cause and effect trials under placebo-controlled conditions, which I'm helping my colleagues do, but we don't have results from that just yet. So that's metformin. You do need a prescription, and some doctors will tell you, absolutely not, I'm not going to give it to you until you get type 2 diabetes. But there are other doctors who are more sympathetic or who have read the research. So that's just a forewarning. The other thing you mentioned, Carolee, is NMN. NMN is a class of molecules called NAD precursors. I talk about them in my book extensively. We've researched them for about a decade. The way to think of these molecules is that they are the gas pedal for the sirtuins that we work on. So sirtuins are defensive enzymes against aging. We have 7 of them in our bodies. And NAD, the NAD molecules that we're taking, the precursors, they're the gas pedal. And resveratrol, which you also mentioned, no, I got that wrong. I should know this by now. So the NAD is the fuel, the gas in the tank, and the resveratrol is the accelerator pedal. And they both work on the same system, on the same proteins that defend the body. And so that's why I think the combination might be helpful. NMN is a molecule that is naturally occurring, and the body converts it into this NAD. What's NAD? Well, NAD is a molecule we need for life. We have a lot of it in our bodies, and it's used for chemical reactions, but it's also used by these sirtuin defenses that we work on as an indicator of when times are tough. When times are tough, our bodies fight disease and aging and fight against or fight for longevity. But without NAD or high levels of NAD, they don't work very well. And in fact, the older we get, in many of our tissues, the levels of NAD decline, apparently by as much as 50%, which I predict is going to be a reason why we age faster and faster as we get older, and why we are susceptible to diseases as we get older. So the idea with NMN is that we can raise those NAD levels back up, give the gas to the system that defends us. And in mice, at least, we've shown, and others, that there are remarkable rejuvenation properties in elderly mice. For example, I mentioned Alice in my lab, and others have shown that you can make mice run 50% further by giving them NMN in their water supply just for 4 weeks. And that's all published if anyone wants to check it out. It was in the journal Cell a couple of years ago. So that's NMN. Resveratrol is the molecule in red wine, which we worked on for many years. We still do a little bit of work on that, trying to tease out the minutiae about how it's actually working in the body. But we have a lot of really good evidence that one of the ways resveratrol works is this accelerator pedal on these enzymes. What do? I still take resveratrol. I've been doing that since 2004. I'm still healthy and alive. So an N of 1 clinical trials being successful, but there have been clinical trials with resveratrol. Some have shown no effect. Some have shown positive effects on things like endurance and blood sugar levels. And all trials are done different ways, of course. Sometimes the patients are older, sometimes they have obesity, and sometimes resveratrol is given with food, without food. And resveratrol tends to work in clinical trials when it's given to people who are older, might have a bit of excess body fat, and also when it's mixed with food that helps it dissolve, because resveratrol is like eating brick dust. It doesn't dissolve very well and can just pass straight through the body.
Mix it into your yogurt?
That's what I do. Yeah, so I did that today, just to prove that I'm not making this up. I have yogurt. I like to use Greek yogurt because I find it dissolves very well in that. But because I'm trying not to eat until late lunch or dinner, it's just a couple of spoonfuls to be able to get the resveratrol into my system.
So, David, you're on the cutting edge of all of this, and so you have such obviously scientific insights into it. As a layperson, obviously interested in staying useful and energetic and all these other good things. And well, how do I go about, if I say to my physician, hey, I'm really interested in NMN and I'm really interested in metformin, I think most physicians are familiar with, but some of these other formulas, they're really not. I know that there are a number of companies out there from whom I can order these products, but without medical guidance, it's not such a great idea. It doesn't matter, or there's a pretty standard dosage, or lead me down that path.
Right. It's a can of worms.
Let's not go there.
Well, I'll say some facts. That's fine.
Okay.
I'm always happy to do that. One is, if you see my name promoting something, it's a lie. I spend tens of thousands of dollars every year trying to stop that. Just heads up on that. Don't go by my name. A lot of the claims are not true, and most of them, just say, "Oh, David's research this, David's research that," but we haven't literally proven without a doubt that, say, NMN works in people, though we are testing it right now. If you do choose to take a supplement, supplements are legal and many of them are fine. I think there's no evidence that I've seen that says NMN is going to do harm, though there's still possibility. Resveratrol, I think, is extremely safe, given that probably 1 million or more people have been taking it for a long time and nothing has shown up yet. So it's this risk reward. The older you get, the more risks you're willing to take, right? In terms of, what's going to happen if you don't do anything. So that's the calculation that I make as well. But if you do take a supplement, no matter what it is, look for the 3 letters GMP, which stands for Good Manufacturing Practices. That means that they're at least, they don't have mouse feces in their factory or whatever. So that's a start. But I would encourage you to look for products that are very pure, at least 98% pure. Because some of the products and supplements can be 50% this, 50% that. And just in the case of resveratrol, 50% resveratrol, what's the other 50%? People don't know. And in fact, there's reports of people having diarrhea, and who knows what else could be happening. So those are my tips.
2 other questions. The first is, I was fascinated by the research that you're doing with the 3 blind mice. And from bench to bedside, is this in our lifetime? How quickly is this research?
Gosh, I hope so. Mm-hmm. Yeah, so I've founded a company called Life Biosciences in 2017. We have raised enough money to get through to human clinical trials in the next 2 years.
Awesome.
If it works, we'll know in a few years from now. If everything goes well, of course.
Yeah, I'm sure it'll work, knowing you. Are there commercially available tests to check the methylation age clock or our true biological clock? And is this related to telomere testing? Is it similar?
Well, it's similar in that they both claim to measure your true biological age. Telomeres are more variable, and I would say, my scientific opinion, not as accurate as this new clock, the methylation clock, but they're both helpful. Telomeres, you might all recall, are the ends of the chromosomes that get shorter and shorter as we get older. There are companies that have these tests. I don't have them listed in my brain, but if you Google Horvath or DNA methylation clock, I'm sure you'll find them. And yeah, these have only been around for a year or two. I can't vouch for them, but I'm keen, out of curiosity, to test how I'm doing. I'm sure many of you are. There's one company, and again, in conflict of interest sakes, I do work with this company. It's called Inside Tracker, and I talk about it in my book. I've been having blood tests from them. They can come to your kitchen. You can go to LabCorp or Quest and get a blood test every few months, or every year if you want. And there they do an extensive blood workup that I also share with my doctor. And it's been very useful to track things over time, so I can see what's working and what isn't, or if something's having an adverse effect. And what they also offer that I found useful, and this is not a sell job, this is just me trying to be educational, is that they compare you to hundreds of thousands of other people and tell you whether you're out of range or not for your demographic, your gender, which is often not done by doctors, and also advice on how to correct things that are out of whack.
I feel like I'm on my Peloton. I know that you track your biomarkers. Is that basically what you're saying?
Yes.
Yeah. Well, listen, I could talk to you, for hours. And we did say that we'd come up to Boston. The last time I saw you clearly was at the beginning of this pandemic in a much more beautiful setting, although your setting with your library is fabulous, knowing that it's your basement. I still am very eager to come visit your lab and bring a group of all of us who are so fascinated and interested in the work that you're doing. And we're just so supportive of your willingness to share it with us. And we know that there is always controversial information that's out there, but we're relying on you, David. Look at what you've done with your dad, which is amazing. So I'm going to rely on you for that. And I'm getting close to him, so in age. So you're on my list to be in touch with. So thank you so much for joining us.
Well, thank you. Thanks for the opportunity to speak and to educate. Happy to do it anytime.